Hello everyone, I'm Ms. Davis, and today we're going to be looking into the domains Bacteria and Archaea. These domains are composed of unicellular prokaryotic organisms. Prokaryotic means before a nucleus, and these cells do not have a nucleus or membrane-bound organelles. All prokaryotic cells do share some common characteristics, so let's take a look at those now. Since bacteria do not have a nucleus, the general location of their one circular chromosome, which is what carries their genetic information, is called the nucleoid. The fluid inside of the cell is called the cytoplasm, and in the cytoplasm you will find ribosomes. Ribosomes are going to make proteins. And also in the cytoplasm, you'll find smaller circular DNA structures called plasmid. The cell is surrounded by the plasma membrane, and this is going to regulate what gets to come in and out of the cell. Outside the plasma membrane is a cell wall. This cell wall offers protection. The cell wall in bacteria cells are made out of peptidoglycan. However, the cell wall in archaea bacteria is different. Outside the cell wall, some bacteria have what we call a capsule, which can help protect the bacteria from their host defenses. Some bacteria also have a flagella, which are tail-like extensions, and these are used for movement, so they whip back and forth to allow for movement. Others have shorter structures called pili, and these are used for attachment. These are these hair-like extensions that extend out. Now, bacteria can have three basic shapes. The first shape that you can see here is cocci, and this is round. So they can also be named like with coccus. The second one here in purple is the bacilli. These are the rod-shaped. And then last are the spirilia, which are the spiral-shaped. So bacteria can come in these three basic shapes. Now there can be modifications on these shapes, however, these are the three basic ones. Prokaryotic cells reproduce by the process of binary fission. Before the cell can divide, it must first replicate its DNA, which you see here. DNA replication begins at one location as the DNA double helix will unzip, and both sides of the DNA get copied. The two chromosomes then attach to the plasma membrane. The cell then begins to elongate. It starts to get longer, and it starts the splitting process. As it starts to elongate, the chromosomes move to opposite ends of the cell. The cell then begins pinching inward, and as the cell pulls apart, this is called cytokinesis, where it's actually splitting. Finally, the new cell wall will form around the two new cells. Binary fission is a type of asexual reproduction, which means that these two new cells are identical as the parent. So they look exactly like the cell they started with. Well guys, this doesn't allow for any kind of variation. Asexual reproduction doesn't allow for this genetic variation in bacteria, but in order for bacteria to survive, they need a way to evolve. Well, they have three ways that we're going to discuss here where they can transfer DNA from one bacteria to another. The first is called transformation. This takes place when a bacteria picks up pieces of DNA from its environment, from its surroundings. And then it begins using it. So you can see here, one cell dies and it releases pieces of its DNA, and then the recipient cell just starts picking up pieces of the DNA and uses them, incorporates them. Another type is called conjugation, and this happens between bacteria cells, where one cell will donate a plasmid to another. And so it offers a piece of its DNA, not all of it, but a piece, and it will actually connect through what we call a conjugation pilus, or sometimes known as a sex pilus. The third type is called transduction. Transduction is when a bacteriophage, the virus we learned about in the previous lecture, transfers DNA from a donor bacteria cell to a new recipient bacteria cell. Now, all three of these processes allow for these bacteria to evolve and change, but this is also how bacteria has become antibiotic resistant. One bacteria is resistant and it shares those pieces of DNA so that other bacteria become resistant. This is creating a problem for us because now we have superbacteria that are not touched by any of our antibiotics that we use today. Now, prokaryotic cells have energy requirements, and there are several different ways that bacteria can process their nutrients. Some bacteria are unable to grow in the presence of oxygen. So if there's oxygen present, they actually die. These are called obligate anaerobes. They cannot be where oxygen is. Most cannot grow, though, in the absence of oxygen. They're like us, and they need to take oxygen in to their cells. These are called aerobic bacteria. The last group can grow in the presence or the absence of oxygen. These are called facilitative anaerobes, where they can switch back and forth between the two processes. Besides the presence or absence of oxygen, some bacteria are considered what we call autotrophic and others are heterotrophic. Autotrophic bacteria have the ability to produce their own organic nutrients or food. Some bacteria are photosynthetic, and they use the energy from the sun to convert carbon dioxide into sugars, just like plants do. An example of this type of bacteria is what we call cyanobacteria, which used to be misnamed as blue-green algae. Other bacteria can do what we call chemiosynthesis. This means that they remove electrons from organic compounds for energy. These are going to be found where there's no sunlight, so like the bottom of the ocean or things like that, but they still need to be able to do a process of making their own food, being autotrophic. Most bacteria, however, are heterotrophic. This means that they have to take in their organic nutrients. More specifically, a lot of them are going to be what we call saprotrophs. Saprotrophs means that they break down dead organic material, so they're going to help in the decomposition process. Domain bacteria includes free-living bacteria as well as pathogenic bacteria that cause many human illnesses. Many species invade and destroy the tissues of their host, while others produce powerful toxins that are harmful to the host. Here is a short list from your book of some human bacterial infections. So here with the sexually transmitted diseases, you see things like syphilis, gonorrhea, and chlamydia. Respiratory diseases, you see strep throat. That's why it's located here in your throat. Tuberculosis goes deeper into your lungs. Pneumonia, whooping cough, and even anthrax. You've heard about anthrax before, like where it was transmitted in the mail and it got mailed to the Pentagon. You breathe it in and it makes you sick. Skin diseases can be like boils. Even acne is an example where bacteria gets in there. Leprosy as well as an example of a bacterial infection. You can see here we also have some digestive diseases like dysentery and cholera. Food poisoning is an example. Nervous system is things like botulism, tetanus, again leprosy when it goes from the skin to the nerves, and spinal meningitis. Because tetanus, you've heard about this before when you've had to get a tetanus shot before. If you step on like a rusty nail or something like that, tetanus will attack your nerves. Okay. Other diseases are like plague, like the Black Plague or the Black Death, typhoid fever, diphtheria, okay, and also like something like Lyme disease. Lyme disease is carried by ticks. So these are a good group of examples. Now, unlike the viral infections, bacterial infections are not prevented by vaccines, but they can be treated with antibiotics. Antibiotics only work on bacterial infections, not viruses. But also we can prevent some bacterial infections again with public awareness. Okay, letting the public know that these diseases are out there. Sometimes when there's like an outbreak of salmonella, they let the public know that you need to be careful with certain products, or they'll recall them so that way that it can help protect them. Hand washing is one of the best ways to prevent the spread of bacterial diseases. But also vector control. This is where we watch what can carry these different types of diseases, and you get rid of them like with ticks or things like that. Domain Archaea consists of bacteria that live in extreme environments, such as hot springs or thermal vents and even salt basins. Since these bacteria live in extreme conditions, they have structures that have modifications that allow for survival in these unique areas. Some examples of Archaea here are included here. These are the methanogens, which are found in anaerobic environments where there's no oxygen, such as swamps, marshes, and even the intestinal tract of your intestines. Okay, they create gas. Okay, so they're methanogens. The second group here are the halophils. These live in high salt concentrations. Now this salt means that there's like 12 to 15% salt. Now this is way higher than even the ocean, and this would be examples of like the Great Salt Lake or even the Dead Sea, as well as what we would call salt ponds. The last group are what we call thermoacidophils. They enjoy extremely hot and acidic conditions. This would be like in hot springs or geysers or hydrothermal vents inside volcanoes, things like that. So they like high heat and they like high acid. So please make sure that you're reading your chapters, and I will see you in chapter 17. If you have any questions, please let me know.